DNA replication is the process of making a new strand of DNA. First, the enzyme named helicase splits the DNA in half. The nucleotides are now exposed. Then, floating nucleotides bond with the original nucleotides with the help of an enzyme. The new strand of DNA is half new, half old, and is called semi-conservative.
DNA translation is a process of using the codons on the existing mRNA strand to create proteins. A codon is a group of 3 nucleotides that is used to help build a protein. The codon AUG is used to start the process of DNA translation. The ribosome uses the codons to create the proteins that are needed. tRNA or transfer RNA is used to transfer the ribosome to the next part of the mRNA strand. Genetic engineering is a manipulation of DNA to give an animal a certain characteristic. To manipulate the DNA of animals for research is just wrong. Overall, genetic engineering is inhumane because it manipulates the DNA of animals to give it a different characteristic than it's originally supposed to be by nature.